Hey guys! So today we are looking at sections 9.2 and 9.3, which are all about photosynthesis, which is kind of the main objective of chapter 9 anyway. Just as a fair warning about these two sections, I'm combining them in your textbook. They have one section, I think it's 9.2, that is kind of like the overview of photosynthesis. And then they go into 9.3, which is the detailed processes involved in photosynthesis. For me, in my mind, at least when I teach this, it makes more sense to kind of do it all at once. So as we're talking about the general kind of pathway, we'll also then touch on the details involved in this process. Okay. So this is going to be a pretty long screencast today. All right. Which is why I've assigned it over the course of 2 days. And if you're in my class, it would take us 2 or 3 days worth of lecture and discussion to kind of get through all of this. So give yourself some grace with this today. Maybe spend some time working through the screencast and take a break and come back and maybe finish it tomorrow. It's on the agenda for both days, so please don't feel like you have to watch this entire screencast today. Okay. So go ahead and get the heading though, sections 9.2 and 9.3, photosynthesis. All right. So we are going to look at the details of this process. I know you all have learned about photosynthesis in the past, but my guess is that you have not learned about it at any level of detail that's going to be similar to what we'll be talking about today and as part of this chapter. So there's a whole lot of information here, probably a lot of things that you're reading here on this objective slide that sound like they would be new or maybe kind of complicated or would include some vocab that you've maybe not become familiar with yet. So again, that's our purpose. That's what we're going to do, is spend time going through all of these objectives that you see right here. Okay, so I'm not going to read through all those today because I have a lot to say otherwise. So we'll just move onwards from that. Okay, so kind of starting back to what we did back in ecology. So thinking about what we know about photosynthesis already and what we've covered so far this year, one thing that we know is that organisms that we call photoautotrophs are those organisms that are self-feeders that do so using photosynthesis. Okay, now this is, by the way, again, this is a review slide. If this is something that you feel like would be beneficial to jot down, by all means put that down. You could at least put down the part up here about photoautotrophs. Like, I feel like that would be appropriate, especially because that's where we're headed here in this chapter, and that's our main focus. But this isn't something that we generally all would write down, okay? So producers that are autotrophs, they make their own sugars, right? And if you're a photoautotroph, that would include plants and protists like algae and kelp. Those do photosynthesis to create their own food, okay? So the picture you're seeing here, they kind of look like blue and orange and green Pringles or something like that. Those are called diatoms. Those are protists that live in freshwater environments like creeks and streams and lakes and ponds. And you can see their colors there. They are colorful because they use sunlight to create their own energy. So those greens, blues, and kind of orange and pink colors you see there are all the colors that they're kind of reflecting. We'll talk about what that means here in just a second. Okay. So thinking about kind of grounding ourselves here as we're talking about photosynthesis, that's just something you're going to want to kind of keep in mind. These are the organisms that we're specifically going to be looking at. So when we look at plants specifically and their ability to do photosynthesis, they have organs that allow them to do that. And one of those organs that they're going to be using are leaf structures. Okay, so leaves in a plant are optimized for photosynthesis. And within those leaves, there are kind of substructures, smaller organs and tissues that play more specific roles than to just say photosynthesis, right? One of those structures is called stomata. Okay, so the first bullet here, you definitely want to jot down. Let's look at some of these other ones. This one, and then this one. So when we talk about stomata, these are structures found on the underside of plant leaves. So, and I wish I had a leaf here with me, but I don't. If we had a plant leaf, as it's sitting along a branch of a tree or a flower, under that plant leaf, so down here beneath the surface, so what's not exposed to sunlight down here, this is where we find our stomata. Stomata look like what you're seeing in this image here. They're called little mouths. Stome was a Latin root and it meant mouth. So stomata roughly translates into meaning little mouths. Now these little mouths do something similar to what our mouths do, which is that they can take in air and they can also release air. Specifically what they're going to take in is one of the reactants that we need for photosynthesis, which is carbon dioxide, CO2. And then one of the products of photosynthesis, one of the gases that gets released as a byproduct here is oxygen. And they'll also release that through these little mouth structures, through these stomata. In addition to that, stomata also can result in transpiration, which is something we talked about back when we did our nutrient cycles, like our biogeochemical cycles. And we mentioned with the water cycle, where you have evaporation and precipitation and condensation, there was this added piece here of transpiration. And transpiration, what it does is it's essentially evaporation from plant leaves. So it's liquid water kind of turning into a gaseous form and evaporating and going up into the atmosphere. That also happens through these stomata. So these stomata can move all sorts of gases into and out of the plant. And when we talk about photosynthesis, this is where we're actually obtaining our CO2. This is where we're actually releasing our oxygen. Now, the one bullet here that I'm not having you write down is about something called guard cells. And guard cells surround these stomata. Guard cells play a really important role. They don't necessarily protect them, but what they do is they help to either allow the stomata to open or to close. And what we know about plants of almost any type is that during the daytime, stomata tend to be open. So the guard cells kind of deflate, which allows them to open up. And at nighttime, those guard cells kind of inflate, which actually kind of closes the stomata. And if you think about why that might be important and why that happens day versus night, well, all it takes is a consideration of what's happening with the plant during the day, right? If there's sunlight, we can expect that our plants are probably doing photosynthesis. And so if we have sunlight and we're going to do the process of photosynthesis, then it makes sense that we also need some carbon dioxide as part of that. Okay, so they open during the day to absorb CO2 and release oxygen, and then they close during the night when there's no sunlight, and when they cannot, well, they could absorb CO2 and oxygen, but at that point, if they were open at the nighttime, they couldn't fully complete the process of photosynthesis, right? So better to be open when you have all the reactants that you need and the sunlight that you need to be able to do the full process, okay? All right, so, oh, and I should also mention one more thing. Those stomata that are at the underside of leaves, it's important that they're on the bottom and not on the top, because if they were on the top, they would suffer more damage from things like maybe insects or rainfall and sunburns, if you want to think about it that way with a plant, right? They're exposed to more potentially harmful things if they were on the upper side of those plant leaves. So that's why we see them at the bottom, okay? They don't actually absorb any sunlight, right? They're just there for gas exchange. All right, so when we look at photosynthesis, again, another structure in addition to our stomata that we use for this process are going to be the chloroplasts. So again, we're looking at the leaves, and you can see I have a picture of a leaf right here. And within that, we're focused on one specific cell. And then within that one cell, we're looking at one specific chloroplast here. And I know it's a chloroplast because I know I'm looking at a plant cell, and I can also see that it's green. It's the only organelle that we've got that actually kind of takes on this green appearance. So chloroplasts are found, remember, in only eukaryotes. These are not found in prokaryotes, though there are some prokaryotes that are photoautotrophic. And in that case, they just don't house these light-absorbing pigments that we're going to talk about right here in an organelle. They're just out in the cytoplasm, okay? But in eukaryotes like plants and algae and other types of organisms that are photoautotrophic and have membrane-bound organelles, we house these light-absorbing pigments in our chloroplast, okay? So light-absorbing pigments include specifically one that we call chlorophyll, which if you've ever heard of a light-absorbing pigment, this is probably the one you've heard about. Chlorophyll's job is going to be to absorb sunlight. And the light that it's actually optimized in terms of really absorbing well is going to actually be red and blue light, which is kind of interesting. In the past, you might have learned that when we talk about light energy, the wavelengths of light that are actually absorbed are not what we see when we look at someone, all right? So, for example, this towel here is kind of this mossy greenish color, right? Well, to me, to my eyes, it appears to be green. And the reason why it appears to be green is because that's actually the wavelengths of light that it's reflecting. So what we see when we look at something is not the color of light that's being absorbed, it's a color of light that's actually being reflected. This is also part of the reason why on a hot summer day that's also very sunny, it's usually a bad thing to wear a black shirt or black clothing. And the reason for that is because that absorbs every wavelength of light, and all of that energy of light is going to be converted into heat into that clothing. And so it actually feels hotter when you wear it. That's in comparison to something like that would be white, right? Like if I have this Kleenex here, that's white. This appears white to me because it reflects every wavelength of light. And so for me, it just appears this brightly colored object, okay? But if it was black, it absorbs everything, and I truly can't even see any colors. It just looks dark to me. So chlorophyll makes the chloroplast appear green, but it's not because it actually absorbs green light. It's really good at absorbing visible light that would technically be in the red and blue kind of spectrum. Okay, so chlorophyll absorbs red and blue, and then it's going to reflect green in terms of its colors. Now, interestingly enough, as we're here in this fall season and leaves start to change colors, what we see is that leaves actually go from being green to yellows and oranges and reds and eventually brown when they kind of die off and fall off the trees. But the reason why we see those different and kind of really pretty and unique colors is because chlorophyll starts to get broken down. So plants, much like other organisms, can tell when there are changes in the seasons and when temperature generally is getting cooler and you're moving from summer to fall to winter. And so what they'll do is they start to break down all those molecules that they had been producing in their leaves that help with photosynthesis, including chlorophyll. And when they break down chlorophyll, we finally get to start to see these other pigment molecules that also play a role in absorbing sunlight. Those are called anthocyanins, which kind of give plant the yellowish color, and carotenoids, which gives the leaves a carrot-like color, carotenoids, carrot, like orange, okay? And those pigments, when they start to shine through, is what actually gives us those cool colors in the fall leaves that we see. Once those get broken down, then that's when the leaves eventually fall off the trees and turn brown and then kind of decay. But the reason why those appear to be different colors again is because they're different pigments. They absorb different wavelengths of light, and they would reflect those oranges, yellows, and reds that you actually get to see. Okay, kind of cool and definitely appropriate, I think, at this time of the year. And by the way, I did not mention this, but of course, I would jot down most of these things on this slide, especially this final statement down here with the 2 vocab terms. Okay. All right, so if we think and dig in a little deeper here to our chloroplast, all right, there are different components, different materials that make up a chloroplast. And so looking at this, these would be some things that we could label. So if you want to kind of sketch a simple diagram of this, that would be maybe helpful. We're going to talk about these specifically with the stages and the actual steps of this process of photosynthesis. So you're going to hear me say these things again. But so here's my chloroplast. This right here is my membrane. Remember, it's a membrane-bound organelle, so you can see it on the outside. And then within that, we have these stacks of what look like and what we kind of think of as like coins or little pancakes. They're usually called one of the two. And so usually when I talk about these, I tend to talk about them like green pancakes, okay? Probably because I like to eat, and I especially like to eat carbs like pancakes. So when we look at these stacks, there's a couple different terms we can give to them. So these are called thylakoids, and this is the actual term for the membrane itself. So when we say thylakoids, those are the individual membranes, okay? So like the membrane here is a thylakoid, and the membrane here is a thylakoid. And you can kind of see these are sliced open here in this example. As if we cut them in half, you can see kind of into the inside of that. They're pretty hollow there. So the thylakoids, when you take them and you stack them all up, we say that they create a granum. Okay, so granum here is a stack of thylakoids. Stack of thylakoids. And when I say thylakoids, again, I'm talking about basically a stack of membranes. Okay, whoops. All right, so this is my stack of thylakoid membranes. So thylakoids taken together are the individual pancakes. You stack them all up. You get one kind of tower, one stack of pancakes, if you want to think about it that way. And that stack of pancakes is what we call a single grana. If you have multiple stacks, like you see in this image, then I have grana. Okay, so granum or grana kind of mean the same thing. Stroma is the other part of the chloroplast. Stroma is the fluid. Okay, so this is the fluid, or you can think about it as like the syrup that's kind of surrounding your pancakes, right? This fluid, however, is not green. All right, not green. And the reason why it's not green is because it does not contain any chlorophyll. So when we think about what's actually happening in the chloroplast, the part that's truly green, and you can see this in the image, is going to be the thylakoid. The thylakoid and the granum are the only areas that actually contain chlorophyll. So chlorophyll. So that chlorophyll is only present in the thylakoids and then those stacks of thylakoids that then make up the grana. Okay, so the part that's going to rely upon absorbing sunlight is going to have to be taking place in our thylakoid membranes because that's where we find our light absorbing pigments. Fluid, the stroma that surrounds these grana, that's also found within this chloroplast but is not green, is where we're going to do really the second step of photosynthesis, which is going to be the part that does not require any sunlight. Okay, so this becomes important as we start to outline those steps. Okay, so when we talk about photosynthesis here specifically, there are really, in terms of this school year, there are only 2 equations that I need you to know, that I definitely need you to know and to memorize, and one of them is photosynthesis. The other one is cellular respiration, and so we'll come back to that when we get into chapter 10. But for this, yes, definitely, you're going to want to jot down this top bullet here that kind of defines what photosynthesis is, and then you're definitely going to want to write out the equation that you see down below. And I've added those green and blue labels there to help kind of emphasize to you where these different reactants and products are being used and then created. Okay. So in terms of the overview, when we think about photosynthesis as a whole, we're using sunlight, using solar energy to convert water, carbon dioxide into sugars and oxygen. And when we talk about sugars, the main sugar we're always going to be referring to here is glucose. Okay, so that's your C6H12O6, and I'll write that here also. So this is glucose. Now, obviously, you're not going to have to write for me this actual equation out. If you do, it would be on an assignment or something else. When it comes to a test, what I would expect is that you can pick it out of, a multiple choice answer option list. So if I give you 4 answer options, you need to be able to pick out the one that does represent photosynthesis. Okay. So water and CO2 are considered low energy reactants. Glucose is a high energy product. And glucose, if you remember from what we talked about in 9.1, is going to be our main source of energy for making ATP. And we'll look at that as we get into cellular respiration in chapter 10. But glucose is really the main purpose for doing photosynthesis. It's not to make oxygen. Oxygen is just nice to have, and we know for living things like us, we require it, and so that happens to work out really well. But the purpose of photosynthesis is not to make oxygen, it's to make glucose, okay? So this equation is sunlight plus 6H2O plus 6CO2 plus C6, or produces C6H12O6 plus 6O2. So lots of 6s in this one. And if you were to count up the numbers of hydrogens and carbons and oxygens on either side of that yield arrow in the middle there, you would see that they're all equal and balanced. Okay. So my light-dependent reactions are going to be my first series of steps that has to occur in order to kickstart this whole process of photosynthesis. And then once we get through the light-dependent stage, we're going to go on and into the light-independent stage, which is going to kind of finish it up. And we're going to end with that high-energy glucose that, again, is the primary purpose for even doing this process. Okay. Sunlight, by the way, I should also mention, is sometimes not even represented in this equation. Sometimes you may get an equation that says H2O plus 6CO2 produces glucose and oxygen, and that would be perfectly accurate as well. Okay. Also remember that my reactants here on either side, so these reactants here or my products here, can actually be switched sometimes. So I could have 6CO2 plus 6H2O produces 6O2 and C6H12O6. Like, it doesn't matter what the order is on either side of that yields arrow. The catch is that water and CO2 stay on the left. Glucose and oxygen will always be on the right because they're always products. Okay. So do add those labels down below where it says light dependent, light independent. And then we're going to go through these stages kind of separately starting right now. Okay. So my light dependent reactions, as I mentioned, are going to occur first. On this slide, by the way, I would jot down all of these. We're going to just kind of do a general overview right here and look at this image, and then I'm going to talk about each process separately and with more detail. Okay. So photosynthesis, again, two different stages of chemical reactions. They rely upon each other. The first one has to occur in order to get the second one to start, and so on and so forth. So the reactants of one are going to be used as, products of the other, and vice versa. The first stage, again, is called light dependent. So light dependent kind of implies that we're dependent upon sunlight, and this first stage definitely is. We need sunlight in order to start this process. If we're talking about this happening at nighttime, we're not going to get this photosynthetic series of reactions to even start because we need sunlight to start the whole thing, okay? If we don't have sunlight available, we're not going to do any photosynthesis. So light-dependent reactions are first, they require sunlight. They take place in the grana or the thylakoid membranes, okay? And remember, that's going to be important here because these are where we actually have our chlorophyll. Okay, so both of these are going to happen in the chloroplast, but my chlorophyll is found specifically, remember, in those grana or the thylakoid membranes, those green stacks of pancakes. The light-independent reactions will follow, and those are going to occur in the place where we do not have chlorophyll. So this is going to be in the stroma this time, that fluid that surrounds the chlorophyll. So you can see kind of in this image, I have one stack of what look like thylakoid membranes. And on this half of the diagram, I've got light-dependent reactions labeled. And then on the other side, I have light-independent, which we're labeling here, occurs out in the stroma. So you can see we have some molecules here that are transferring from the thylakoid over to the stroma. And then we have kind of opposite named molecules going backwards, all right? And this is where I'm saying that these two are reliant upon each other. They're linked because of these molecules that move from one stage to the other, or one location in the chloroplast to the other, okay? So looking at light-dependent reactions first, okay, if we had to kind of summarize them in just a statement, this is what I would make that statement look like. So the light-dependent reactions use sunlight and water to produce energy carriers for the light-independent reactions, releasing oxygen as a byproduct. So that was kind of a lengthy statement, but that's again because this is not a complicated or easy process. All right, so really if you want to summarize and capture everything that's happening, you have to be super specific like this. So if you think about light dependent, well, we can identify some things already. We're using sunlight and we're using water, so those must be our reactants. We're going to produce energy carriers, which are thought of as a product in this case, and we're also going to release oxygen as a byproduct, another product. All right, notice I did not say glucose. Notice I did not say anything about carbon dioxide. Those will come later in the second stage. So in this first stage, we're not dealing with anything here that's carbon-based. We're dealing with sunlight, which is not made up of, anything biological that we're talking about. Water, which is hydrogens and oxygens, and then oxygen. So no carbons involved in this first series of steps. Okay, so here is kind of an overview of what we see happening as part of this first step. And if it looks overwhelming, it should, because it is. I assure you that what we're going to do with this first step is to focus on the most important aspects, okay? So you're going to see lots of detail, but I want you to think about, okay, the summary of this all, right? The big picture. So think about, why is this important? Try to keep that in the back of your mind. How does this happen in general? But don't worry too much about getting hung up in the details, okay? By the way, if you're going to pause and come back to this later, this would be a good stopping point because I'm going to just talk through the steps here and that'll be the rest of this screencast today, okay? So light-dependent reactions consist of groups of molecules that are embedded in the membrane, so not in the cell membrane, but in the chloroplast membrane, in the thylakoid membrane specifically within that space, called photosystems. So photosystems, photo was a Latin root, it means light. These are systems of proteins that absorb light. All right, and you can see them here in this thylakoid membrane. All right, and I know it's a thylakoid because it's labeled down here, and I can also see that it's green. These photosystems contain lots of chlorophyll and other types of light-absorbing pigments, and you can see, they're representing here the light kind of coming into this space. All right, these different wavelengths of light. So the photosystems, their main job is to absorb sunlight to kind of start this process. When that happens, this is going to be kind of the series of events that will occur in response to that. So photosystem 2, which is kind of ironic, photosystem 2 starts this process. Photosystem 1 actually comes after. But photosystem 2 is going to start this process of photosynthesis. And what photosystem 2 does, this is where we'd have photosystem 2, by the way, is going to be in this general space right here. This is PS2. And that photosystem 2 is going to absorb sunlight, and as it absorbs sunlight, it's going to immediately use that energy to split a molecule of water. All right, so down here, this H2O molecule gets split, and I should probably scoot back and not circle that like that. Okay, so my water molecule gets split. That's the very first step. Absorb sunlight, split water. Notice when I split water, I get 3 different products. I get 2 electrons, I get 2 hydrogens, and I get half of an O2, which is like one single oxygen atom. But we know oxygen doesn't want to sit by itself. So it's going to wait around until it gets a binding partner, another oxygen, to be released as oxygen that we breathe in. Okay. So the 2 electrons that get released from this H2O molecule that got broken apart, those 2 electrons are going to start moving through something called the electron transport chain, or the ETC. So here are my 2 electrons. They're going to come up here to my ETC, and they kind of start to bump through this chain of proteins that really is sitting in the membrane there. Now, they're not representing those there for you. You can see the phospholipids that make up that thylakoid membrane, but they're not showing you those proteins, okay? But these electrons will start bouncing through these proteins, and as they bounce through, the hydrogen ions that we also released through the splitting of water right here are going to come up here, and they get pumped across the membrane. So these are ions, so keep in mind those are things that can't normally cross these phospholipid membranes on their own, so they're going to need help from transport proteins. And in addition to that, this is an active transport process, which means it's going to require our new favorite molecule, ATP. So we're going to have to move them across the membrane, and we're going to transport them in such a significant way that they don't necessarily want to go that way, okay? That there's going to end up being a very high energy or high concentration of these hydrogens on the opposite side of the membrane inside the thylakoid. And that's going to be something that we know these ions wouldn't naturally want to do on their own, okay? So these are my first 3 steps involving this first stage. So again, chlorophyll in photosystem 2 absorbs sunlight that splits water. As a result of that splitting, oxygen can be released. 2 electrons move through the ETC, and the energy from those electrons bouncing through the proteins of the ETC is used to actively transport or pump hydrogen ions against a concentration gradient across that thylakoid membrane. Okay, if that sounds complicated again, it is. Okay. All right, so then we move on and we have a second photosystem. Okay, similar proteins, but it comes after the first photosystem. Now, we call the second photosystem photosystem 1, which again, I said is like totally weird because it's coming second. The only reason why it's named photosystem 1 is because it was found first. Okay, and then they didn't go back and actually change those names. But photosystem 1 comes after photosystem 2. Like photosystem 2, we're absorbing sunlight. And as we do that process, we're going to re-energize our electrons that have been bouncing through this electron transport chain. All right, so this is kind of, and that's not yellow. I meant to make that yellow here. Have been bouncing through that ETC. Okay. At the end of that ETC, what we end up seeing happen as a result, these electrons have to go somewhere. All right. They can't just float around in a cell without actually interacting with something else. So those energized electrons bounce through those ETC proteins to the point to where they eventually reach the end. And at the end, they're used to create this molecule down here, which is called NADPH. So my 2 electrons get added to my NADPH, to NADP, made into NADPH. And NADPH is an energy carrier, all right? That energy carrier's job is going to be to kind of transfer the energy that's held in its bonds, the energy that was added by those energized electrons coming from the water that was split by the absorption of sunlight. It's going to take that energy and it's going to transfer it on to the second stage, to the light independent reactions. Okay. So NADPH will go on to the next stage, and notice where we're at. We're already out in the cytoplasm. Sorry, not out in the cytoplasm. We're already out here in the stroma, where we said we're going to do the next stage anyway. Okay. The thylakoid's up here, and then the inner chloroplast and thylakoid is in this space. But this is my stroma. This is my fluid that surrounds my thylakoid. All right, so that's what I've kind of got here. That NADPH will go on to the next stage. In addition, I'll also make another energy carrier called ATP, which I'll show you here on the next slide. But you can see here I have ATP being produced. That ATP gets made thanks to this concentration gradient that we've created on the opposite side of the membrane. So remember, we were pumping hydrogen ions across the membrane, right? On the last slide, we talked about that. Those hydrogen ions, notice over here, are in a high concentration, and that high concentration of hydrogen ions will allow it to diffuse back down a concentration gradient. And as that happens, that protein channel helps to create ATP. So the name of this ATP creating protein channel is called ATP synthase, and this is what ATP synthase looks like. Okay, so the image here up at the top is like a computer model of what this looks like based on the components that we know make up ATP synthase. This one here at the bottom is an actual picture taken with a microscope of what ATP synthase looks like, which, if you can imagine, we're talking about a protein embedded in the thylakoid membrane, which is found in the chloroplast, which is found in a eukaryotic plant cell. Like, you're talking about really tiny things, right? So that's why it looks so fuzzy. But we know ATP synthase kind of functions like a little propeller. So here's my ATP synthase. So as we're kind of finishing off this first stage, where I've got lots of hydrogen ions on one side of the membrane, what ends up happening is these can diffuse again down a concentration gradient to the other side. And as that happens, as they go through this ATP synthase propeller, the actual propeller itself kind of starts to spin around in like a clockwise fashion or counterclockwise fashion. And as a result of that, that spinning is what we use to add a phosphate to ADP. That's not a D. ADP to make ATP. All right, and then that ATP, like NADPH, also functions as an energy carrier and will take energy from this first stage, light dependent, to the second stage, light independent. Okay, so this in this case is due to this, passive transport or diffusion of hydrogen ions that causes this protein molecule to start spinning and turning. The creation of that hydrogen ion concentration gradient is because of the energy provided by the splitting of water and that absorption of sunlight. Okay. So in reality, what we're getting out of this, we got an NADPH molecule, and we also got ATP. All right, and here is kind of our summary image of what's happening. So again, we absorb light here. This is our first step. Light gets absorbed here, and as light is absorbed, we split water. That water releases electrons that will go through the ETC. So here's my ETC. My photosystem 1 here absorbs more sunlight. That does not split more water, but it re-energizes these electrons, which need a place to go and eventually get added to NADP plus to make NADPH. The hydrogen ions that got actively transported across the membrane and down their concentration gradient, like you can see here, are used to then drive the production of ATP as they flow back down from high to low concentration across the membrane. Okay, the only thing I'll add about this is that this is kind of flipped. On the last couple images I showed you, the thylakoid space was up here above, and the stroma was down here. And now we've kind of flipped that image upside down. Okay, that's why it looks like hydrogen's pumping the opposite way. It's not, it just kind of depends on which area of the membrane you're focused on. Okay, so kind of big picture ideas. All right, first stage, absorbs sunlight, splits water, releases oxygen, and creates energy carriers. So again, that's NADPH over here and ATP over here. And notice both of these go on to the light independent reactions, okay, which again are going to be happening out in the stroma. All right, so here is that second stage, the light independent reactions, which by the way are also called the Calvin cycle. All right. We call these light independent because they do not require sunlight. They can happen with or without sunlight. It's just not a necessity. All right, which is also the reason why we're talking about this happening in the stroma where we have no chlorophyll. So these can actually happen at night. Long time ago, when scientists were first trying to identify all this stuff, they kind of inadvertently called these the dark reactions because the assumption was that they happen in the dark. They can, but they don't always. All right, so that's why we've renamed them to be light independent reactions. So their main purpose is going to be to use the energy carriers produced in the first stage here, NADPH and ATP, and create glucose. All right, so what we're going to get out of this is going to be high energy sugars like glucose, and also oxygen will be released. Oh, sorry, excuse me. Not oxygen will be released. We just did that. So we're going to make glucose, and as part of that process, we also need to use some CO2. My mistake. All right, so remember, this point, we're going to start using some carbon. We have carbon in CO2. We're going to have carbon in glucose, which remember is C6H12O6. But we're not liberating or creating any other byproducts. Okay, the main purpose, again, is going to be glucose specific. So light independent reactions, unlike the light dependent. Here we occur in the stroma. Again, we do not need sunlight, and it can occur at night. So all those things that I just mentioned, if you didn't jot those down, I would go ahead and do so here. You can see on this diagram, this is a picture of what we call our Calvin cycle and the light deep independent reaction stages. So notice it's a cycle, much like any other cycle we've seen this year. There are multiple steps. Really, it's much more involved than even this. There are multiple enzymes that catalyze multiple reactions and so on. This is, again, about as simple as we can make it, okay? So, no, you don't have to diagram that whole cycle. You don't really even have to know that diagram except to say, where does it occur and kind of the general pattern and the general pathway that's taken by this, okay? So in terms of the steps, I have just 5 steps. I want to make sure I didn't say the wrong thing. 5 steps here for our light independent reactions. So maybe in some ways this might be simpler than what we saw, with the light dependent reactions, the first stage. So we start with CO2 in this case. We again have 6 molecules of CO2 that we're going to be using, but in reality, we're just talking about, hey, what would happen if I just start with 1 CO2? So ATP and NADPH energy is going to be used here at this stage to start rearranging and breaking apart and creating new molecules from the carbons that we obtained from CO2. So for every 1 glucose, which is C6H12O6, we need to have 6 carbons, which means we need 6 total CO2 molecules. So those 6 CO2s come in, they start to be rearranged and recombined in different ways. You can see my ATP here coming from my light dependent reactions is going to be used, as well as my NADPH, which is used. And they're both used in terms of like when we break those bonds found within those molecules, either between the 2nd and 3rd phosphate of ATP or between the hydrogen and the phosphate of NADPH. That's going to provide energy for us to rearrange some of these molecules. So we go from a 3-carbon molecule here to another 3-carbon molecule here. They both have some different names, but again, they're super complicated, so we're not going to worry about that right now. And then we rearrange enough to where when we combine two of these 3 carbon molecules, we end up with our sugar. We end up with our glucose. Okay, so our 6 carbon sugar, aka glucose, is created by kind of joining 2 3 carbon molecules to make that ring-shaped glucose structure. Okay. There are carbons that are left. They continue to be rearranged. We continue to invest some more energy in order to create those other molecules. And then again, whatever's left and remains goes back through this process and repeats. Okay. So it is a cycle again. There's no start, there's no stop. But kind of the limiting factors, if you want to think about it that way, would be the ATP and the NADPH that you absolutely need in order to invest to start kind of moving all these pieces around, okay? If you lack the ATP and NADPH from the light dependent reactions, if you never produce them there, if you don't have sunlight and you can't go through that process, well, then you're not going to be able to make glucose because you absolutely need energy coming from that first stage, okay? So that's light independent, also known as the Calvin cycle. So here's kind of the same summary image. It's a little bit bigger, so maybe you can see it a little bit better. Again, kind of the same idea as what we just talked about in the last slide. You don't have to rewrite this whole diagram. I care more that you have kind of the big picture idea down and not so much the individual details, all right, if that makes sense. So don't stress about this whole process, but understand, okay, where is this happening? What's required? What's produced? And why is it important? Those are kind of the main ideas that I always tell kids to kind of focus on. So in this case, light dependent, let's think it happens in the stroma, which is part of the chloroplast. It's using energy carriers and carbon dioxide, and it's producing glucose, right? Why is it important? Well, because it produces glucose, and it's the final stage of photosynthesis, okay? We could do the same kind of series for the light dependent reactions also. All right, so moving back, hold on, I went one too far. Last little part of this section is looking at factors that can affect the rate of photosynthesis. So obviously, you know, the amount of sunlight we know can affect how photosynthesis occurs. In general, the higher the light intensity, the higher photosynthesis, the faster that rate becomes, the more photosynthesis can be done, the faster the growth, and the more glucose. Same with availability of water, right? The more water, the more you should be able to do this process. In temperature, generally, as we go up in temperature, we also increase our rate of photosynthesis. Now, that's to a certain point, right? If we get too hot, then we don't have really the ability to do that, okay? The last little piece right here, there are some plants like cacti and like the plants you see here, like succulents, that have the ability to live in extremely bright and very hot conditions, okay? So what these guys tend to do, these can be called C4 or CAM plants. You don't need to know much about them, but interestingly enough, you know, the stomata that we talked about earlier that I said open during the day and close at night, these plants have adaptations that allow them to only open their stomata actually at night so that they can prevent water loss. And what they do at that point is they bring in the CO2 that they need and they start to kind of incorporate that into molecules in the Calvin cycle, but then they kind of wait for the energy carriers coming from the light dependent reactions and they can create those during the day. So they kind of separate photosynthesis into this two-part event, whereas in most plants, it kind of occurs consecutively. In plants like cacti that live in desert-like conditions where they run the risk of being damaged or potentially dying from water loss, they'll make all their energy carriers when it's sunny outside and during the daytime, and then again, they'll wait until the nighttime to open those stomata, absorb CO2, and kind of finish off the process. So it's a really kind of unique way for those plants to be able to survive in those types of harsh kind of conditions. Okay. So that's all for 9.2 and 9.3. If you've got any questions, as always, let me know. Go back and review this if you need to. I know I've got some assignments coming up here that will also review this whole process with you. All right. So again, focus on the big picture items and continue to review. All right. And I will see y'all later. Bye.